Welcome viewers. Today in this session I'll be talking about a very interesting physiological phenomenon occurring in all of us, that is DNA replication. So let us see what do we understand by DNA replication. DNA replication, as the name suggests, the DNA molecule replicates. The DNA molecule after replication or after being copied to its progeny strands transmits the genetic information to the progeny cells during the cell division. There are a number of enzymes involved in DNA replication. One of the major enzymes or one of the most important enzyme is DNA polymerase 3. Apart from this, there are other accessory enzymes helping out this important enzyme. Now we will see what are the detailed mechanism of DNA replication occurring in both prokaryotes as well as in eukaryotes. Now before we cover all of these topics, let us first watch a beautiful animated video on DNA replication showing the process of DNA replication in details. I think you must have enjoyed this video and it will be very interesting when you will study the mechanisms more in details. So let us now proceed to the detailed mechanism. So before that, let us first know what is semi-conservative mode of DNA replication. As the name suggests, the semi-conservative mode of DNA replication means that the two strands of the DNA double helix unwinds to form the DNA template. As we can see in this slide, the DNA templates serves as the base on which the new nucleotides are synthesized. So they are acting as the templates. So these are known as, or this is known as, the semi-conservative mode of DNA replication. Now let us look at the wonder enzymes. What are the enzymes which are helping in the process of DNA replication? The main important enzyme is DNA polymerase. Apart from this, there is single strand binding proteins known as SSB and as well as helicase. So all of these enzymes are important for DNA replication. DNA polymerase, it's something like a boss kind of an attitude, and it is the main enzyme which carries out the polymerization as well as elongation of replication. Then apart from this, the two DNA strands, when they are acting as templates, they are covered by single strand binding proteins, which prevents these strands to become re-annealed. Then we have the helicase enzyme, which is acting at the replication fork and causing the untwisting of the double helix. So let us see how all of these enzymes acts like friends together and help to carry out the replication process. So this picture shows how the DNA helix present at the replication fork is unwinding the double helix with the topoisomerase acting ahead of the replication fork and removing the supercoiling or positive supercoils. Now the DNA polymerase are of very importance, as I have told you earlier. The main kind of DNA polymerase found in prokaryotes are polymerase 1, 2 and 3. Now polymerase 1 was first isolated by Arthur Kornberg. Later on it was found that polymerase 1, when treated to proteolytic treatment, it resulted in the formation of a large fragment, or a large fragment was obtained which was known as the clenofragment. The clenofragment has its application in molecular biology. Returning back to the previous slide, so the other types of polymerase are polymerase 2 and 3. Polymerase 2 is mainly involved in DNA repair, whereas the main enzyme acting in replication is polymerase 3. All of these enzymes have the ability to polymerize from 5' to 3' direction, as well as an exonuclease activity present in polymerase 1 from 5' to 3', as well as from 3' to 5', and only in polymerase 3 the exonuclease activity is present from 3' to 5'. Knowing about the DNA polymerases, we should do some justice to eukaryotes also. So the types of polymerases in eukaryotes are mainly alpha, delta and sigma. Apart from this, there are other polymerases like beta, eta, theta, all are involved in the DNA repair process. Apart from this, again there is another polymerase called gamma present in mitochondria. Now, what is the basic common property of all of these polymerases? All of these polymerases helps in the polymerization capability. That is, they have the polymerization ability. What does it indicate or mean? Let's see this slide. What can we see? That there is a template and there is a growing DNA chain. At the 3' end of the DNA chain, the incoming nucleotide is added. So, the polymerase enzyme is capable of adding nucleotides only to the 3' end of a growing nucleotide chain. So, this is conserved for both prokaryotes as well as in eukaryotes. Mechanism of replication. There are 3 steps: initiation, elongation and termination. Now, all of these 3 steps are somewhat similar in prokaryotes as well as in eukaryotes. Let us first see at the prokaryotic model. So, if we look at the prokaryotic model, we find that the initiation of DNA replication starts at a particular point. This particular point is known as ORI C. On closer examination of this ORI C, it shows it is 245 base pair long, consisting of repeats which are 9-mer and 13-mer. The 9-mer sequences are present in 4 copies. The 13-mer sequence, rich in AT, is present only in 3 copies. So what if it is present only in 3 copies? So let's see further. On further examination, it is found that the DNA A protein is able to recognize the 9-mer sequence. It binds to it, followed by the DNA B binding to the ORI site. The DNA B, which is a helicase and is delivered to the origin of replication by DNA C. DNA B binds to the 13-mer, unwinds the DNA double helix as shown in this slide, and as a result, the closed complex formation changes to the open form. Now, when it changes to the open form, the 2 DNA strands thus become single-stranded and are covered by single-strand binding proteins, and they can now serve as templates for DNA initiation. Now, this slide shows how it initiates. If we look at this, so first the primase enzyme binds to the 5' end, initiates a short RNA primer. To the 3' end, now the DNA polymerase 3 is able to add the nucleotides to make it grow. In this way, the initiation of DNA is dependent on a RNA primer. On closer again of examination of the ORI C, we find that the double-stranded molecule becomes denatured to form a replication bubble. The replication bubble consists of enzymes as well as the single-stranded DNA templates. Now further on, it is found in both the cases, that is in both prokaryotes as well as in eukaryotes, the DNA replication is of bidirectional type. If we look at this picture, we can find that there are 2 Y replication fork or 2 Y-shaped structure which are connected at their tops to form a replication bubble. This is known as bidirectional replication. So this is very important feature of DNA replication. Now, how the DNA polymerase 3 attaches itself to the DNA? It is with the help of a sliding clamp. The sliding clamp first attaches to the DNA molecule, and it is very much similar to the rods on a curtain. So as a result, it is able to slide over the DNA. Now how does it get attached to the DNA or the clamp gets attached? It is with the help of a clamp loader complex which uses the ATP hydrolysis to load the clamp on DNA. Further on, the DNA polymerase III is made up of plenty of protein subunits, or it is a multi-subunit structure. The holoenzyme consists of a core enzyme and 20 other accessory proteins. The core enzyme of DNA polymerase III is made up of alpha, theta and sigma. So all of these three subunits makes the DNA polymerase III core enzyme, which is able to add nucleotides. Now, if we look into this picture, we can see that the DNA polymerase III is able to act only in pairs. So both the core assemblies assembles one on each strand, and the two polymerase III are connected to each other by means of a tau subunit. Now let's come to the initiation process in eukaryotes. Now there is a remarkable feature about eukaryotic replication. In eukaryotes, the DNA replication only takes place in the S phase. What is S phase? S phase stands for synthesis phase. So it is only in this phase that the DNA replication is occurring. Whereas in prokaryotes, DNA replication occurs whenever the cell is grown in nutrient-rich medium. Now we know eukaryotes contains more DNA than prokaryotes. As a result, there must be multiple origin of replication rather than a single origin of replication to replicate or copy all the genetic information present in eukaryotes. As a result, we can see multiple origin of replication, and the sequences present in origin of replication are less well characterized than prokaryotes, and these are known as autonomously replicating sequences in or of eukaryotes. Let us see, so the initiation, so similar to the prokaryotes, first the DNA untwisting takes place with the help of DNA helicase, and the single strand binding proteins binds to the single stranded DNA templates, after which a primase sits at the 5' end, initiates a short RNA primer. The short RNA primer is further extended by DNA polymerase alpha or it produces a DNA of 3 to 4 base pair long. This is known as initiator DNA. The initiator DNA is then bound by replication factor C. The replication factor C then attracts the pCNA bound with the polymerase delta, and then the polymerase delta extends the growing polynucleotide chain. In this way, the initiation of DNA replication in eukaryotes takes place. Now, let us see or compare both the eukaryotic and prokaryotic initiation. This is a very simplified diagram showing the initiation. So, if we look at this diagram, then we will find that this is the replication fork. The replication fork is moving in a definite direction. Now, since there are two strands or two templates, so it is giving rise to two DNA molecules, newly synthesized DNA molecule. Now, one DNA molecule or one strand is being synthesized continuously, moving in the direction of the replication fork, and this is known as the leading strand. Whereas the other strand which is moving opposite to that of the replication fork and is known as the lagging strand. Moreover, if you look at the enzymes are DNA helicase, exactly acting at the replication fork and thereby unwinding the DNA double helix. Along with the primase enzyme synthesizing the RNA primer, SSB protein binding to the two templates, and so on. Further on, if we closely examine the lagging strand, then we see that the lagging strand is made up of discrete series of DNA fragments. So this means a lagging strand is not synthesized continuously. Now, before we go further into the lagging strand and its interesting feature, first see what is DNA looping. So the DNA looping, as I told you earlier, that the two DNA polymerase III, they are supposed to remain in or they remain in pair. So to remain in pair, now since one of them is acting in a replication or one of them is acting in a direction opposite to that of the replication fork, so there must be looping of the DNA. The looping of the DNA enables the DNA polymerase III to be paired to each other with the help of their tau subunits. So who loops? It is a lagging strand which is producing the looping. Or if there would have been no looping, then the 2 polymerase III subunits would have moved apart from each other. Let's take up the Okazaki fragments or the lagging strand. As the lagging strand consists of a series of discontinuous DNA synthesis. On this closer examination, it was found this short DNA fragments are known as Okazaki, named after its discoverer. How are they synthesized? First, this is the parental DNA, which is masked with single strand binding protein. Then Pri-A protein binds and displaces the SSB protein from the template. After it has displaced, it allows the primase binding. Primase initiates a short RNA primer, and then DNA pol III sits and adds nucleotides to the 3' end of the RNA primer. Thus, this kind of a structure or a short Okazaki fragments are produced on the lagging strand. Thus, the Okazaki fragment contains DNA with gaps in between them. In order to have a continuous strand, the gaps has to be fulfilled or has to be sealed. So, there are 2 enzymes operating now. One, DNA polymerase I, another is DNA ligase. DNA polymerase I removes the RNA primer present at the 5' end of each Okazaki fragment. It replaces the RNA with DNA. But still, there are gaps in between the short DNA fragments. These gaps are covalently sealed with the help of DNA ligase. So, thus producing a continuous duplicated DNA strand. Now the DNA are replicating, so it is now entering the elongation phase, both in prokaryotes and eukaryotes. As we can see, the elongation is taking place in the direction of the replication fork, continuously untwisting and fork movement, thereby causing the templates. The templates are separating out, and then the DNA pol III is synthesizing or adding nucleotides to the end of the growing polynucleotide chain. Till how much it proceeds? Until and unless it ends the end of the DNA molecule. Till then, it is under the elongation phase. Elongation in eukaryotes is slightly different. Here, it requires the presence of 2 enzymes, DNA polymerase sigma, DNA polymerase delta. Now, DNA polymerase delta mainly does DNA synthesis of the lagging strand, whereas DNA polymerase sigma, it causes continual DNA synthesis on the leading strand. Similarly to the prokaryotes, there must be helicase, which is continuously untwisting the double helix, a topoisomerase acting ahead of the replication fork, and in this way again continuous DNA synthesis in the leading strand, this continuous DNA synthesis in the lagging strand, and then we have the ribonuclease. The ribonuclease removes the RNA primer present at the 5' end. The ribonucleases which removes the RNA primer are ribonuclease H1 and ribonuclease FEN1. Apart from this, since the eukaryotes, they lack DNA polymerase I, thus the RNA primers, though are removed from the 5' end, but the gap are not able to be resealed back because the eukaryotes do not have DNA pol I. Now, this causes a problem which I'll just discuss a few slides later. So, this is all about the elongation of eukaryotes. Now, the elongating or the eukaryotes, since it is present in the form of linear chromosome, so this is continuously happening in the 2 directions till it reaches the ends of the chromosome. Let's see how the termination. So, the DNA is replicating and now it has reached to the ends. This signals or triggers termination. How it triggers? Now, the termination in prokaryotes, again if we look into this diagram, we find there are TER sites. These TER sites are nothing but they are the termination sites present, and these are 22 base pair long. These sites are recognized by TUS monomers. Now, since there is a bidirectional replication, so the two replication fork are moving in two different directions. In order to prevent reduplication of the already duplicated genetic material, the termination or the replication fork should terminate at the proper position. For this, there are the TER sites. Now, these TER sites prevent the further extension of the replication fork after it has reached the TER sites. In this way, they prevent the reduplication. The parental strand, or we know that the parental DNA, it gives rise to two new circles, or the two newly synthesized DNA. The two newly synthesized DNA are held to each other and they are separated by means of topoisomerase 4. Thus, the topoisomerase 4 helps in the separation of the daughter newly synthesized DNA strands and passes the genetic information correctly to the daughter cells during the cell division. So, in this way, the entire process of replication is completed in prokaryotes. Now, in case of eukaryotes, there is a problem. Since, as I already told you, that the DNA replication in eukaryotes is occurring in the form of linear chromosomes. Now, the linear chromosomes contains at its 5' end RNA primer. The RNA primer, after it is removed, it leads to the shortening of the chromosome as there is no enzyme which can fill in the 5' gaps. This is known as end replication problem, which does not occur in prokaryotes as it is a covalently closed circular DNA molecule. To solve this problem of termination in eukaryotes, there are telomeres. The telomeres found at the ends of the chromosome, they help in the termination of transcription as well as they help in maintaining the length of the chromosome, which would have decreased if the 5' primer or the 5' position would have shortened with every cell division. DNA replication is a marvelous feature of living organism. It cannot be accomplished without the help of certain enzymes which act together like friends and help to complete the process of replication. It is through this process of replication that the genetic information is passed on as it is from the parental DNA to the progeny DNA. Thus, with this, DNA replication is an important feature of the central dogma and without which this living organism would have come to a halt.